So how do we help the public differentiate between a paranoia about pharma that relates to misprescription, overprescription, overpricing, all of these socioeconomic challenges? How do we help people separate that from a paranoia about science and what molecules are and what they do and what a disease is and things that are really not disputable in the physical realm?
Yeah, well, it's difficult, right?
Yeah.
So all modern like pharma drug development is based on science, right? And that leads to some of these very high drug prices, right? So
High risk.
it's very high risk.
Put
right?
a lot of money in. You're trying to
You
make a
have to put a
drug.
lot of money in.
Doesn't work. You lose all that money. So pharma has a bit of a higher profit margin than other industries, right? What would you say that is, roughly?
I mean, it's hard to say
Right.
on the drugs because it's very difficult to know like what really went in there. And drug development is such an expensive, time-consuming process, right? So if you have a disease you want to treat, it isn't just like, oh, okay, you want to cure lung cancer or something like that. You have to find the specific one. You have to validate what you want to target, right? So you have so many of these proteins in your body that if you inhibit other ones, that's how you can get bad side effects and like leading to death. So you have to validate your drug target, and then that's what starts the drug development process, and that can be 20 years.
You have to figure out a molecule that will inhibit this protein, and then you have to make sure it doesn't inhibit anything else that you don't want it to.
And that's what takes the 20 years is the trial.
That's what can
And
do
then,
it.
and then speaking from the perspective of someone who did some organic chemistry, you actually have to figure out a synthetic pathway to mass produce this molecule, which can be super duper challenging.
Yeah, no,
Interesting.
yeah,
It's like not enough just to figure it out
Right.
that one time.
You then, if it's something that you've isolated from a natural source, well, maybe it only comes in these microgram amounts in a sponge at the bottom of the ocean. We got to make this thing in a
Yeah.
lab so everybody can use it.
Like crops. It's like you don't just grow one lemon.
Right? You got to
Yeah.
grow, you mass produce this molecule, right?
No, yeah, and I think one good example of that is like turmeric. So turmeric has curcumin in it, which I've done some work on like amyloid diseases, like Alzheimer's. It's like the main one of that, and curcumin, like people find it all the time, like it's going to inhibit like Alzheimer's, like aggregation in your brain, right? So it sounds like, oh wow, great drug, right? You can go ahead, put in a pill, give it to people. Sure, that molecule in the lab works, but then
In a biological system,
it doesn't
there's
work.
a lot of factors.
You have so many factors. So like once you identify what you want to target, you have to screen millions of compounds. And so if curcumin is your compound that comes out, that might not be a drug-like compound. So it may not work in biological system, can have off-target side effects. It can maybe it doesn't have good bioavailability. So you take a pill and then you just can't absorb it.
Yeah, just piss it out.
Yeah, just piss it out. Or it only accumulates in the wrong tissue, not to the right levels, has crossed the blood-brain barrier. There's so much to develop in that. So you have to make those drugs. You have to, after you find your initial hit, you have to go through organic synthesis and make, hundreds, thousands of other compounds and hope they maintain the activity and have better properties or improved specificity over another protein or what. But you can't predict that. Right now, we can't predict that. So it's a lot of empirical work, trial and error to find what you want. And hopefully you achieve that, in a reasonable time frame.
Mm-hmm.
But all easy drug targets have really been screened already. So currently we're in this state where there's so much competition and secrecy of what's going on. You may know what a rival company is working on, but you don't know how far they are. So you can put in 10 years of effort to get something.
Oh my God! And these guys could be wrapping it up.
They could be wrapping it up, or you could be ready to go into a clinical trial.
Hence, patents are so important.
Yeah. You can be going into a clinical trial, and then the patent come out that claims the same molecule or like a class of molecules.
At what point do you patent? Do you do that before the trials, just in case?
So it would be before the trials, but I mean, it's a difficult difficult space because when you have a patent, you only have it for a few years.
10 years or something? How long is it?
I believe 20 years.
Does that
total.
make it one of the first things you do then? Just like, well, I should probably give us 10 years.
It is
to develop a little bit more of this.
No. You want to do it as late as possible.